Alrighty, welcome back everybody. We are going to cover the final chapter in Unit 1. And this material will be the final material that will be covered on the exam this week. We're continuing with cell biology, and this should say cell bio part 2, and it is chapter 5. So basically what we'll do is just start off with a little bit of review what we talked about before. Remember, we were talking about the organelle, and we got into the cell. We talked about the cell theory, okay? We talked about the anatomy of the cell in the previous chapter. What are we going to focus on with this chapter is the cell boundary, the cell membrane, aka the plasma membrane, okay? So remember when we talked about the phospholipid molecule under the macromolecule structure of lipids, and we said that the phospholipid bilayer is responsible for the plasma membrane, and the function of the plasma membrane is to separate internal and external environment. That means to separate what's going on outside of the cell from what is occurring within the cell. So remember that when we talk about the structure, we have a fluid mosaic model, and it's called a fluid mosaic model of the phospholipid bilayer because it is in actuality a fluid membrane. It is dynamic. It is constantly changing and fluctuating. It is not like a wall in your rooms right now, or a wall that you're sitting in your living room, bedroom, dining room, listening to this lecture right now. It is actually a dynamic structure that changes and adjusts according to the environment and adjusts according to the needs of the cell. So the fluid mosaic model is comprised of the lipids. Remember, it has cholesterol within it, the phospholipid, which is the phospholipid bilayer, proteins, which we're going to talk about in great detail right now. So remember, we went over that under macromolecules, and then carbohydrates. In terms of the function of the biological membranes, you have obviously structural support, but you have also the function of transporting substances into and out of the cell. And we're going to talk about how that occurs. And then finally, you have the function of communication. Yes, cells will communicate with one another. All the cells in your body, believe it or not, communicate with one another. There's a communication system, an internal communication system within you that helps to identify cells and also tells certain cells where to go in the event of injury. For example, when you cut yourself and the platelets rush over to that injured area and start the healing process. All right. So if we take a look at the picture of the phospholipid bilayer, you can see there's a lot going on here. And this is actually a remarkable structure. First, you have the phospholipid molecule, okay? So we have our hydrophilic head, our hydrophobic tails. We have these little blue guys embedded in there, and those are the cholesterol molecules, and those are only found in animal cells. We have the external, we have glycoproteins, we have an integral membrane protein, also known as a transmembrane protein. We have peripheral proteins that are found on the interior of the cell. So this is the outside, so that's why we're calling it the extracellular environment. So all this stuff is outside of the cell. And then this is all inside of the cell. So there's a cytosol. Remember, the cytosol is the water-like fluid found within a cell. Now, if we take this phospholipid molecule and we magnify it, okay, you can see this is the outer part of the cell, the extracellular. This is the intracellular portion. And right here you have the phospholipid molecules within one another, and then you have cholesterol that's found in between there. Cholesterol functions to help maintain the fluidity of the membrane. That helps to ensure that the membrane stays fluid. That means movable, okay? It means dynamic. Remember that the non-polar tails are going to interact with one another because they're not going to interact with water. And then the polar tails will interact with water. So they're going to interact with extracellular fluid, so the fluid outside of the cell, which again is comprised of mostly water, and then the intracellular fluid is going to interact with the hydrophilic heads on this side, okay? So really amazing picture right here, figure 5.1, showing you the remarkable structures that are involved in the plasma membrane. So I mentioned the membrane proteins. It's one of the first things that we're going to talk about. Membrane proteins. You have two types. You have integral proteins, and then you have peripheral membrane proteins. The integral proteins are transmembrane proteins. Trans, across, membrane. So it's crossing the membrane. So it is a protein that has communication with the outside of the cell, and a protein, the same protein, has communication within the cell. Pretty remarkable. You also have attached to the integral membrane, you have the peripheral membrane proteins, okay? The membranes are semi-fluid, so we said they're fluid, but they're not completely water-like, okay? They are actually semi-fluid, which means that they can move within one another, and there's lateral movement, okay? And that helps to allow the passage of substances, okay? So we're gonna be talking about that. The movement of the membrane depends on the composition of lipids, okay? The shorter nonpolar tails, the unsaturated fats, and the cholesterol will help to ensure that the membrane remains fluid, okay? And then you have what's known as lipid rafts, and that's where you have a high number of cholesterol, and that can lead to serious problems in animals, okay? So with the movement of substances, so we talked about the transmembrane, AKA integral proteins. The function of those proteins is to help transport molecules across the membrane. This is why the plasma membrane or the cell membrane is described as selectively permeable. It allows the passage of certain substances, but not all substances. And it will allow the passage of substances based on their polarity, based on their size. So that helps because you don't just want stuff going into your cells and causing harm to your cells. Because if something foreign gets into the cell, remember that you can disrupt the function, okay? So let's talk about the substances that are highly permeable, okay? Gases can cross the membrane readily, okay? Carbon dioxide gas, oxygen, obviously, you know, nitrogen gases, even the bad gases such as carbon dioxide, nitric oxide, hydrogen cyanide gas, all those can cross membranes. Alcohol, ethanol can cross the membrane very easily. It doesn't need the help of any transmembrane proteins. So the substances that have high permeability are very small and they're uncharged molecules. And basically we call that the gases and then the other substances. For example, alcohol is a really small molecule and it can cross. The molecules that have moderate permeability, okay, so they do need some help. And we have what's known as aquaporins, which were discovered in the late 60s that help water cross. So water, we want to be able to cross the membrane very easily, okay? But also urea, for example, a substance that we need to be able to break down, that's produced by metabolic waste. Low permeability molecules are going to be, for example, polar organic molecules such as sugars, okay? So glucose, even though it's super important, and we're going to be talking about it a great deal, it cannot just readily cross the membrane. It needs help. Finally, you have very low permeability substances. And we describe these as ions or charged polar molecules, and then macromolecules, those ginormous molecules that we talked about. So ions, anything in your body that you consume that can become an ion needs help to cross the membrane. Sodium, potassium, magnesium, calcium, chloride, manganese, chromium, iron, all those require a transmembrane or integral protein. What else? How about amino acids that we need to build proteins? ATP that we use for energy. Proteins. Proteins are huge, huge molecules. You can have 4,000 amino acid proteins in your body. Or the polysaccharides. Remember glycogen? Or for example, if we consume starch, okay, 20,000 carbon long molecules. Or what about nucleic acids to cross the membrane? How about 6 billion nucleotides? Yeah, you need something to help it cross the membrane. So we're going to talk about the movement of substances, and we divide the movement of substances into passive transport processes, which require no energy, and active transport processes, which require energy. There are subcategories that you find under passive transport. Those are simple diffusion and facilitated diffusion. So we're going to be talking about all those. So simple diffusion, okay, let's say, hey, we have oxygen. This is all oxygen that we just inhaled. We need it to cross the membrane, and we want it to go into the mitochondria so we can undergo cellular respiration. That's going to be simple diffusion process. You're going from an area of high concentration to low concentration until equilibrium is reached. If you caught that, which I know a lot of you did, you should know that I just gave you the definition of diffusion. Diffusion is the movement of a substance from an area of high concentration to low concentration until equilibrium is reached. In this case, it is the simple diffusion movement of a substance across a membrane down the gradient. So it's moving from high to low. So basically that means you are abiding by the law of diffusion. As I stated earlier, we cannot in biology ever violate the laws of chemistry and physics. So if there's a law in chemistry or a law in physics, we cannot violate it when we're talking about biology. So therefore, the law of diffusion is a physics law, and it's a law that pertains to gases, but then here we can actually attribute it to molecules. So you're moving down a concentration gradient. That's simple diffusion. Facilitated diffusion. What does facilitate mean? It means to make easier, to help, to assist. So you're facilitating diffusion. So that means diffusion is still occurring. That means what? You're moving from an area of high concentration to low concentration. You're moving across the membrane. However, what's helping it, what's assisting it, what's making the substance easier to cross, what is aiding it? You guessed it, the protein. And what does that look like to you? Oh, that looks like a transmembrane or integral protein. Yes, yes, yes. Okay. The third mechanism that we see here in this figure is the movement of a substance from low concentration to high concentration. Wait a minute. How can that be possible? Well, we're moving against the concentration gradient. Now, some of you are going to be like, wait a minute, doctor, you just said that we cannot violate the laws of chemistry or physics. Correct. We cannot. But can you push a boulder uphill? Absolutely you can, but what do you need? You need a tremendous amount of energy, or you need help. For example, you can take a bulldozer and roll it up the hill. You can do that. This is exactly what we're doing here. What we're doing is we're taking energy in the form of ATP and with the help of a protein, and we're going against the concentration gradient. This needs to occur for a lot of the metabolic processes that occur, and actually a lot of the physical processes that occur that you can actually see require active transport. And we're going to talk about that. So there are different cell gradients that are found in a cell, and we know that cells are able to maintain a relatively constant internal environment. That means maintain homeostasis, okay? We want the certain number of substances within the cell. We want them at a certain level, okay? You want that to be completely unchanged as much as possible. You want it to remain constant to the best of the cell's ability, okay? And also, you have to remember that the internal environment is going to differ from the external environment. So you can have two different types of gradients that we see with cells. A concentration gradient, which is the concentration of substances, or an electrochemical gradient, which is absolutely fascinating. We'll talk about a little bit in 181, a little bit in 182, but we'll go into in great detail in Bio 201, Human Anatomy and Physiology 1. Okay, so molecules like space to move. They want to be in equilibrium. Okay, so when we talk about passive transport processes, it's like taking this solute, okay, pouring it into this beaker, for example. So I'll say square the beaker, and you can see it starts spreading apart, and then it's all equal within the beaker. The example I like to give is back when I was a kid. I remember the hot days of summer, growing up in Vermont and hanging out with my friends when we'd go to each other's houses. And the best part would be the lunchtime breaks that we'd have after playing outside. And our parents would make a pitcher of Kool-Aid, and we could watch it being made sometimes. And we'd argue about who'd be lucky enough to be able to rip open that Kool-Aid packet and pour it into that nice big pitcher of cold water, and immediately pour it in, and you see the powder just go into the liquid, and all of a sudden you see the clear water become red water, and then you see it all equal, and then mom or friend's mom mixes it, and then you pour the glasses or the cups for all of us, and we all have the same great tasting Kool-Aid, right? What was that? Little did I know at that time, yep, that was just a basic diffusion process, simple diffusion, area of high concentration to low concentration until equilibrium is reached when you pour in that packet of Kool-Aid. Now, with active transport, you're producing a gradient and you're disrupting the equilibrium. You're going against it. And that needs to happen in order for physiological processes to occur in the human body. Again, you can see molecules of dye, okay, pores going through, and then diffusion across, and then equilibrium. You have equal amounts on each side of the membrane. Yes, the membrane. Okay, so area of high concentration to low concentration until equilibrium is reached. So you have equal amounts in the extracellular fluid, and then you have equal amounts in the intracellular fluid. Okay, equilibrium, equilibrium. All right, cell solutions. You can see a solution is what? It's a solute plus a solvent. So the inside of the cell, we describe it as intracellular solution or fluid, aka the cytosol, which is just the fluid by itself, or the cytoplasm. That's all the organelles and proteins and everything that's found in there, and amino acids and sugars. That's the cytoplasm. On the outside of the cell, you have the extracellular solution or extracellular fluid, and then you have interstitial fluid, which is basically another name for extracellular. It's found between the tissues, okay, which we're going to get to later on. All right, I want to talk about a really important diffusion process, and that is the process known as osmosis. Osmosis is basically the process of diffusion, and it's the process of facilitated diffusion, because as I explained earlier, you need aquaporins to cross the membrane. Why do you need aquaporins for water to cross? Think about that for a second. Why would you need an aquaporin, which is what? Yes, you guessed it, an integral or transmembrane protein. Why would you need that to cross the membrane? If you answered because of the hydrophobic tails in the phospholipid bilayer, you're absolutely correct. So aquaporins are the specialized transmembrane or integral proteins that are responsible for moving water in and out of the cell. All right. Another important concept is tonicity. And tonicity has to do with the way water moves. Water in a biological organism, in a living organism, moves due to the solute concentration. We have three different scenarios that can occur with the movement of water. And this occurs in everything from a single-celled organism, such as a paramecium or an amoeba, or a bacteria such as E. coli or staph or strep, to you as a 100 trillion cell organism. So we have three different possible scenarios that can occur. You can have a isotonic solution in which the concentration of solutes is equal to the solvent. So basically, what you have is the concentration that is found in the cell is going to be equal to the concentration of solute outside of the cell. Okay, so that means, oh, there's not going to be any movement of water. Water's happy. It's at equilibrium. So the concentration of solute is equal in the extracellular fluid, and the concentration of solute is equal in the intracellular fluid. Pretty easy. Iso meaning the same. So if you see isotonic on an exam, which you probably won't ever see, but it's going to be the same because that's too easy. That's why you don't see it on my exams, and you probably won't ever see it on a standardized exam. They just don't ask that. Okay. What they do ask you about is the other two scenarios. First one we're going to talk about is a hypotonic solution. So if you place a cell in a hypotonic solution, that means that you have more solutes inside of the cell as opposed to having the concentration of solutes on the outside of the cell. So basically, what that means is that, wait a minute, I am sitting in a hypotonic solution. So what that means is I have more solute inside the cell than I do outside the cell. What do you think is going to happen to water? Well, you come back up here and you see water follows solutes. So this is the first thing I remember on my first week of clinical rotations, and my first clinical rotation was in surgery, general surgery at West Penn Hospital in Pittsburgh, Pennsylvania. And at the end of our first week, usually on Friday afternoons, we'd have meetings where the chief resident would give a lecture. And I remember something, and I remembered it to this day, and I tell my students every single semester. And he told us a very simple way to remember how disease occurs. Basically, he stated that water follows salt, sugar, and protein. So whenever there's a higher concentration of salt, sodium chloride, sugar, glucose, or protein, in this case, we talk about the interstitial protein, albumin, then water is going to follow that, and that's going to lead to a disruption. Well, what happens to the cell when it is placed into a hypotonic solution? When the cell is placed into a hypotonic solution, the water is going to go into the cell and cause the cell to swell up, fill up with water, and eventually burst. So that is the second scenario, the hypotonic solution. Now let's say that we place a cell into a hypertonic solution. What's going to happen? Well, in a hypertonic solution, hyper meaning more, tonic, the solutes, you have a higher concentration of solutes on the outside of the cell than you do on the inside of the cell. Therefore, where's water going to go? Right, water is going to move out of the cell to try to make the reach equilibrium and make the concentration equal. Therefore, what's going to happen to the cell? It's going to lose water. It's going to shrink. And that's very, 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 very bad. Okay, so 3 scenarios that are very important that you need to be aware of. All right, so when we're talking about it with different types of cells, whether it's animal cells or plant cells, the best example that we use in biology and in physiology and anatomy and everything is the example of the red blood cell. Because the red blood cell is found in abundance in the human body. So basically, your red blood cells right now, okay, if you're sitting and you're listening to my lecture, I'm absolutely 100% sure that your red blood cells are in an isotonic solution, okay? If there was a fluctuation, you would be ill, okay? So if we change the concentration, okay, and we place the red blood cell, let's say that we increase the salt concentration in our bloodstream, okay, that means in our plasma. Well, water is going to leave the red blood cell, and that's going to result in the shrinkage. In case of red blood cells, we call it crenation. Crenation, okay? Now let's say we do the opposite scenario. We place the red blood cells in a hypotonic solution. So we cut your consumption of electrolytes. That means you have no sodium, no chloride, nothing. And you're just drinking water, water, water. Well, the concentration of solutes, for example, sodium and chloride, are going to be higher in the cell than they are in the plasma. Therefore, water is going to go into the cell, cause it to swell, and then eventually, boom, the cell explodes and it's dead. This is called hemolysis, the bursting of a red blood cell. In plants, it's a little bit different because remember what we have in plants. What do we find in plants that we don't find in animal cells? What do we find in plant cells? Yes, what am I pointing to? The cell wall. The cell wall prevents, okay, major changes in the structural integrity of the cell, the plant cell. Okay, so even if you get a lot of water or you get a little water, yes, you will have the increase in water if you place the cell in a hypotonic solution, or you have the decrease in the amount of water if you're placed in a hypertonic solution, okay? But the plasmolysis and the expansion of the cell is not going to be what? Catastrophic to the cell, because you can see what? The cell maintains its structural integrity. Chloroplasts are okay, nucleus is fine, chloroplast is okay, nucleus is fine, mitochondria is okay, there's mitochondria. Everybody see that? Tonicity, 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 very, very, very important. All right, so you can see the difference between the cells. So here's a cell in, so you just watch this video and you can see the difference between what happened to this cell. All right, so let's talk about the transport proteins. The transport proteins, going back to them again, they are transporting substances. What are they transporting? Sodium, chloride, potassium, magnesium, amino acids, glucose, carbohydrates such as glycogen, starch, whatever it's going to be, okay? Basically, what you're doing is the transport proteins allow for selective permeability. Remember that word, we talked about it a little bit ago, okay? So you want to be able to move stuff in and out of the cells. So we have different types of channels that can be used. We have voltage gated or chemical gated channels, And we're going to talk about that in great detail in 201 and 202. I don't get into it too much in 181, but I do want you to be aware of it. So when you get to that course in the future and your instructor, your professor is talking about gated channels, you'll be like, oh yeah, transport proteins. That means they're moving, they're going to open up, either if they're voltage gated, they're going to open up in regards to a difference in charge, or if they're concentration gated, they're opening up to a difference in concentration. Okay, so you can see the different ones. We're not going to talk about them, the uniporters, symporters, antiporters, don't worry too much about those. Finally, the active transport mechanism, which I talked about, all require energy. So you can have an ATP fueled pump, okay? The sodium potassium ATPase pump is a big example, okay? You can also have the secondary active transport, which is gradient driven, and then you actually have vesicular transport. So then you can have actually vesicles that will transport the substances across the gradient with the help of ATP. All right, there's a sodium potassium ATPase pump. Very big, huge. When we talk about nervous system physiology, you're going to see that the movement of the sodium and potassium going against the concentration gradient using the ATP is going to be very important. Now they talk about the steps here, and it's mentioned in this amazing figure. I just want you to be aware that the sodium potassium pump is an example of an active transport mechanism. So what does that mean? It means that sodium and potassium, those ions are going against their respective concentration gradients. That means they require ATP. And this happened to be the ATPase, okay? So here's a nice video showing that. Again, just be aware of it, okay? Again, you don't know the details of how and what and the number of 3 and 2, so on and so forth, okay? We'll talk about that in Bio 201. All right, so you can see this table showing the important functions of ion electrochemical gradients. Just take a look at that. I'm not going to test you on it, just be aware of it because we will be covering it in future courses. Vesicular transport is basically membrane-bound packages. So again, vesicles will grab on to and then bring in or get rid of substances. So we're going to talk about the big ones that you see. We have an exocytosis process and an endocytosis process. Endocytosis process, you have 3 subcategories. You have pinocytosis, phagocytosis, and then receptor-mediated endocytosis. So with exocytosis, what you're doing is you're taking a substance, okay, and you are releasing it, okay? For example, how do you think we secrete hormones into our bloodstream from our different glands? For example, from your thyroid gland, or from your adrenal gland, or from your pancreas, okay? It's with the help of vesicles. So they're going to carry their hormones, and then they're going to eventually take the hormone and then release it into the extracellular environment. In this case, if this substance in the triangle is a hormone, let's say it's going to move into your bloodstream. And then that hormone is going to cause a specific response. Again, we're going to talk about that in great detail in Human Anatomy and Physiology 2. So you can see there it is, the description, hormone. It's huge. You can see all the hormones are released from your glands via the process of exocytosis. We also have the release of digestive enzymes via exocytosis from our stomach, from our small intestine, duodenum, jejunum, and ileum, so on and so forth, okay? With endocytosis processes, the first subcategory is pinocytosis. And basically what you're doing is you are taking in fluid, all right? You're taking in water droplets and you're bringing them in to a vesicle and then transporting them, okay? The second is phagocytosis, which is pretty extraordinary. You can watch this video right here. A lot of your immune protective cells have the ability to undergo phagocytosis. The big one, which is shown here in this cartoon, okay, going after the bacteria or the protozoa or even the viruses, is Mr. Macrophage right here. So basically he acts like a Pac-Man. So this is the extracellular fluid. The bacteria is floating around in there. It grabs it, engulfs it, and it closes in on it. And then this vesicle delivers this pathogen, or they're calling it the particle here, to the lysosome. And remember, what does the lysosome have? That's right, it has hydrolytic enzymes. It's going to dissolve the bacteria or protozoan or virus in the acid and kill them. All right, so watch this really cool video. And finally, we have receptor-mediated endocytosis. A lot of substances have to move in to cells via receptors. One example is glucose. Glucose moves in via receptors attached to your cells. Cholesterol has to move in to your cells via receptors. Okay, so very, very important process. So what it does is, for example, you have like these triangles. Okay, they are attaching to the receptors, and then, for example, let's say that this triangle is glucose. That means that receptor is insulin. It's going to grab on to the glucose, bring it in, engulf it, and then it's going to use that glucose. The cell's going to use that glucose for what? Yes, we talked about it. We're going to talk about it. We mentioned it briefly. Yes, cellular respiration. Okay, so really important processes right there. Again, you can see the uptake of a lot of substances here that undergo endocytosis. Okay, and you can watch this video here that shows you differences between endocytosis and exocytosis. Another video. All right. So remember to undergo to study the terms, but remember, don't just memorize them. Try to link them together and tell the story. And then also the study guide concept, the questions. Be able to explain this to someone who has no knowledge of maybe science or biology, okay? And tell them, hey, I have a story to tell you, okay? And that's how you're gonna be able to answer questions on the exam. So when you're doing this, remember, as I explained in the other lectures, when you're doing the study guide questions, be sure to use your references first to answer the questions, write them out, make sure you know them, then try answering the questions in the study guide without any reference material. And remember, put this all in your own words to tell the story, okay? So, ladies and gentlemen, that is Unit 1, and that is the material that will be covered on your first exam, which will be this week. Most likely, it'll be on Tuesday or even Wednesday, so that I can allow the Zoom meeting, okay, on Tuesday, because I know that you're going to have questions after this weekend and after going through all the material. All right? Take care, and I will be in touch on Zoom again. If you have any questions about this material, you always know to send me an email or contact me via Zoom. All right, take care, everybody.